Welcome everyone to the Cell Bio section of the Leading Edge Symposium. I'm Claudia Vasquez. I'm an incoming assistant professor at UW, and I'm co-hosting this with Zara Weinberg. She's a postdoc at UCSF, and we're really excited to be moderating the session, which will kind of really span and stretch the idea of what is cell biology, really looking at like neurons and bacteria and different just varying systems of how organelles are organized and allow cells to do the various things that are amazing that allow us to be us. So our first talk is Mónica Quiñones-Frías. Sorry, I just messed up your name. Mónica Quiñones-Frías. And Mónica, if you want to set up your talk. She's a third-year postdoc in Avi Rodal's lab at Brandeis University. She did her PhD in Troy Littleton's lab at MIT. And Mónica, take it away.
Awesome. So hi, everyone. So today I'm going to be telling you about my current efforts to develop tools to visualize neuronal ER, as I'm showing here on this first slide, with the goal to study the structure of the ER in these cells and study functional contacts of the ER with other organelles, in particular endosomes. And so here I'm showing a drawing by Ramon Micajal, and I've always loved his drawings because they capture the beautiful complexity of neurons. And I've always been intrigued by these cells because they contain highly specialized compartments and exhibit unique features, such as generating electrical currents to communicate with other cells. And so throughout my career, I've been pursuing or studying fundamental questions about the cell biology of neurons. And so during my graduate work, I studied mechanisms that regulate synaptic vesicle fusion dynamics. And I discovered that a protein called synaptotactin 7, its levels at synapses tightly regulated neurotransmission. And I also discovered that this protein is not on synaptic vesicles or the sites where vesicles typically fuse, but instead it's on a compartment away from it. And this was really interesting to me because it made me curious about how other organelles at synapses could contribute indirectly to neurotransmission. And so for my postdoctoral work, I decided that I wanted to explore how membrane-bound compartments, in particular the ER, regulate neuronal physiology. And I became interested in the ER because even though it's a highly abundant organelle at synapses, we know very little about its structural dynamics or even functions of it at synaptic terminals. And so I also became interested in how the ER might regulate endosomes at synapses, because work from non-neuronal cells has shown that these compartments are interacting extensively, but we really don't know what their function is in neurons. And so the endosomal pathway initiates by the endocytosis of membrane and lipids at the plasma membrane that then form vesicles that undergo a series of maturation steps that can either lead to the degradation of endosomal cargoes through the lysosome or fusion of these compartments with the plasma membrane and then release of these cargoes through exosomes. And although the ER was traditionally thought to be involved in protein synthesis, work in non-neuronal cells has shown that ER organelle interactions, such as with organelles with like mitochondria, regulate a wide variety of functions. And here I'm highlighting what has been discovered about ER endosome interactions. And in neurons, these ER functions could play critical roles in the physiology of these cells because they are heavily dependent on endosomal traffic for long-range transport of molecules and signaling, as I will explain a little bit later in my talk. But we don't know if these functions that had been discovered in non-neuronal cells are conserved across cell types, or if neurons have unique functions for these ER endosome contacts to meet their unique features. And so to explore these questions, I decided to use the Drosophila larval neuromuscular junction as a model to study neuronal ER. And for my work, I'm focusing at ER at axon terminals, which I'm highlighting the motor neurons here in green in this video of a larva crawling. And so if you zoom further at these connections between the motor neuron and the muscle, you would see that the axon terminals look something like this. And these swellings here are called boutons, and they contain the machinery that's important for neurotransmission. And I will show you that there's ER there. And some of the advantages of using Drosophila as a model is that it's extremely easy to do dissections in these animals to be able to visualize these axon terminals. They are 10 times larger than mammalian central synapses. And I have available a large genetic toolkit that allows me to probe ER function. And so when I started my postdoc, I developed tools and a protocol to visualize with area scan microscopy the live dynamics of ER at axon terminals in Drosophila motor neurons. And to visualize neuronal ER, I expressed in neurons a GFP that has a ER retention sequence. And so it will label the lumen of the ER. And in collaboration with members of the Phi Lab in the math department at Brandeis, I've been developing new computational tools in order to track live the changes in structure or the dynamics of the structure of the ER over time. And the reason why I'm really interested in studying ER structure, is because there's neurodegenerative diseases that have been found to be caused by mutations in genes that regulate ER structure. However, this has mostly been explored in non-neuronal cells. And so today I'm going to be telling you about a mutant that disrupts particularly the formation of three-way junctions, which is this structure that I'm highlighting here in the ER's structure. And this protein is called elastin. And so elastin is a GTPase that mediates the
Interesting.
mediates the homotypic fusion of ER tubules and upon GTP hydrolysis. And so, right. And mutants of elastin have been studied in non-neuronal cells, and what they found is that ER tubules in these cells are messed up. And these phenotypes are primarily observed towards the periphery of cells where ER tubules are predominant. And as I alluded before, elastin is one of those genes that has been previously discovered in this type of motor neuron neurodegenerative disorder called hereditary spastic paraplegia. And so to visualize the ER in elastin mutants, I used the same approach that I described before, where I expressed in neurons this luminal ER marker, and then I image the structure of control or mutant synapses using area scan microscopy. And I discovered that similar to other cell types, the structure of the ER in these mutants was significantly disrupted. However, I discovered that some unique features in elastin knockouts at axon terminals. One of them is that the ER in these cells that lack elastin have a ladder-like ER structure that I never see in controls. And I don't have time to go through this analysis that I did, but I found that the density of ER tubules near the plasma membrane in these mutants is reduced. And then finally, which is the phenotype that I was most excited about, is that it appears that elastin mutants have luminal ER proteins in the cytosol. And this is really interesting because it suggests that maybe luminal proteins at axon terminals of elastin knockouts might be leaking out to the cytosol. And so my working model is that these ER luminal proteins might be leaking out of the ER because the integrity of the membrane of the ER in the mutant is compromised. And so to test this model, I'm planning to label the ER by expressing a GFP that is tagged with a transmembrane domain of an ER resident protein. And if I don't observe cytosolic labeling, it would suggest that the membrane of the integrity of the ER membrane is compromised in these mutants. However, another scenario is that the ER is highly fragmented in this mutant, and at the resolution of our system, it appears as cytosolic labeling, as seen with the luminal ER marker. And so I was interested in further exploring the function of ER structure in these elastin mutants. And so because previous work has shown that they have a wide variety of endosomal phenotypes. And one of them is that the axon terminals of these elastin mutants have this abnormal synaptic growth that has previously described as being due to disrupted growth factor signaling at synapses, which is dependent on this endosomal trafficking that I was describing earlier. And the other phenotype that has been described is that in elastin mutants is that they have an increased number and size of these multivesicular bodies. And as I was showing before, right, endosomes go through these different maturation steps. And one of them is this multivesicular body stage that can then degrade or release cargoes to the outside of the cell, which could indicate that these mutants might have disrupted endosome maturation. And so to explore this further, I decided that I wanted to look at endosomal cargoes that we know traffic through this pathway, where they end up being released to the outside of the cell through exosomes. And the reason is because at the Drosophila neuromuscular junction, when endosomal cargoes are released from inside the cell to the outside of the cell, they become trapped in the muscle. So it allows us to really easily quantify the levels of cargoes inside and outside the cell. And when we do this for elastin mutants, what I observed is that neuronal cargoes seem to increase inside the cell without any change on the outside. And what was really interesting is that I only observed this phenotype in the longest motor neurons in Drosophila, which is in parallel to the motor neurons that are defective in patients with hereditary spastic paraplegia. And so to explain what I think might be going on is that, so I mentioned that as endocytic cargoes are being trafficked through and are maturing, they can be degraded or released outside of the cell. And because I didn't see any change on the levels of endosomal cargoes on the outside of the cell, I think that ER endosome contacts might actually be really important for the trafficking of cargoes that are destined for degradation. And in neurons, degradation is it's known to mostly occur on cell bodies. And so what I think is happening in the mutant is that this transport might be disrupted, which leads to a buildup of endosomal cargoes that should be degraded. And so with that, I want to summarize what I've told you today. So I've developed tools to study ER structural dynamics, and I found that ER structure might be important for preserving ER endosome contacts that are important for the degradation of cargoes. And I found atlastin as part of a screen to identify the effects of disrupting different ER genes at synapses. And I found that others like elastin increase endosomal cargoes, but I also found others that decrease. And in the future, I'm interested in using these new quantitative tools that I've developed to study changes in ER lumen and ER membrane dynamics, and also explore how neuronal activity can regulate structure and ER organelle contacts. And with that, I want to thank my lab, especially Avi, who's an amazing mentor, and the Phi Lab for helping me generate these new quantitative tools to study neuronal ER, and the Leading Edge to give me a platform to talk about my research and then my funding sources. And with that, I can take questions.
Thank you, Monica. That's a beautiful talk. If anyone would like to have any questions, if they want to ask them themselves, please raise your hand. Or if you want us to ask them, please put them in the chat. in the meantime, I had a question. This like ER analysis is really like, incredible that you've developed with the Phi Lab. And I was wondering if something that's kind of like you noticed was that the atlastin mutant is at these kind of tri-ER contacts. And I was wondering if there was any reason as to why, like, if there's something kind of interesting happening at these tri-ER contacts where it's more susceptible to endosomal, like mutations to endosomal cargo, or if it's just like, is that a really dynamic spot in the ER network, or is that a really stable spot in the ER network, and that's why endosomal trafficking is going through it? Like,
so the question is, one thing, I don't have a tool yet to accurately identify ER endosome contacts. I'm actually working on to build a sensor to detect those sites. Actually a few technical challenges. So one, which doesn't allow me to answer this question, because if I express, the answer is no, I don't know. But it's because I not of lack of trying, because if I express markers to label the ER and endosomes at the same time, so and these are overexpression, it actually leads to this crazy phenotype where then you get these huge blobs of both ER and endosomes. So it's definitely messing up these compartments in the cells, but it's something I'm really interested, right? Because having a sensor to be able to determine where these sites are happening in the control, and then seeing in the mutants, like if there's more contacts or less contacts would be great to answer, but work in progress.
cool. Anne Martin says, beautiful work. Do you see the ER present in the post-synapse as well as, and how, do you see this ER present in the post-synapse as well, and how do the dynamics compare?
Yes, so the muscle has tons of ER, which is why I need to like express ER markers only in the neuron in order to be able to visualize it. But I haven't really studied the dynamics in the ER yet. I kind of just quickly expressed it to see that it was there, and it was, and I kind of didn't focus on it. But one thing I want to do, which we'll study this a little more, is since there's this cytosolic labeling in the neuron, right, in these elastin mutants, I want to see if I can see it in other cell types in Drosophila, and I'm thinking of looking at the postsynaptic compartment and see if I see this cytosolic labeling there, and so I would be studying the dynamics then more.
And then I'll ask you Julie Siegenthaler's question. She says, wonderful talk. Julie Siegenthaler from the University of Colorado. Do you think this dysregulation of ER dynamics impacts synapse elimination? Example, more prone to lose the synapse?
I haven't noticed any changes. So when I do the experiments, I don't really notice that there are less synapses, at least in this specific at the NMJ. I couldn't say of others, but at least at this synapse, I don't notice that there's times that I go to that specific muscle and I don't see a neuron.
Thank you so much, Monica. You have another question from Jiyun in the chat. And now I'll pass it on to Zara to introduce our next speaker.
Awesome, thank you.
Thanks, Claudia, and thanks, Monica, for a wonderful talk. It's my pleasure to introduce Lisa Marie Nisbett. Lisa Marie did her graduate work with Elizabeth Boone at Stony Brook University and is now a postdoc in Jessica Seeliger's lab, also at Stony Brook. Lisa Marie is a NIH IRACTA NY CAPS scholar, and today she's going to be telling us about the cell biology of microbes. Lisa Marie, take it away.
Thank you so much. Can everybody see my slides?
Yes, looks great.
Okay, great. So good afternoon, everyone, and thank you to the organizers of this symposium for giving me the opportunity to talk to you guys about one of my research projects entitled Exploring the Mechanism of Lipid Transport to the Outer Membrane of Mycobacteria by the LPRG RB1410C pathway. And so for those of you that are not familiar, mycobacteria are rod-shaped bacteria of the actinomycetes family. Mycobacteria can be environmental and pathogenic, especially the infamous human pathogen Mycobacterium tuberculosis, which is a causative agent of tuberculosis and is responsible for approximately 1.5 million deaths per year. So whether pathogenic or not, mycobacteria consistently have to deal with assaults from the environment, such as exposure to antibiotics and host immune responses when inside the host. So to protect themselves from these environmental assaults, mycobacteria possess a structure called the cell envelope, which is comprised of an outer membrane, a periplasmic layer, and an inner membrane. And the protection that the cell envelope confers to mycobacteria is due to the presence of the outer membrane, which is a very well-armored structure due to the presence of an array of molecules, including lipids that are chemically diverse and extremely hydrophobic. So as you can see, these lipids are not your everyday lipids. And compared to phosphatidylcholine, which is a lipid that's typically found in the membrane of our cells, mycobacterial outer membrane lipids are very large, very complex, and very hydrophobic due to the presence of these very long hydrocarbon chains. So to build this critical outer membrane layer, mycobacteria must transport these lipids from where they're made, which is at the inner leaflet of the inner membrane, to the outer membrane, and must do so by transporting these lipids through the periplasmic region, which is hydrophilic in nature. So to shuttle these lipids as is would be very energetically unfavorable for mycobacteria. And so we think that dedicated lipid transport mechanisms must exist in mycobacteria. But to date, the mechanisms that drive lipid transport in mycobacteria still remain unclear to us. And as a result of that, this means that there's a fundamental gap in our understanding of how mycobacteria transports its lipids to the outer membrane. And this gap, therefore, further prompts us to ask, what is the identity of the lipid transport pathways that exist in mycobacteria? And what are the molecular mechanisms that these transport pathways use to shuttle the lipids to the mycobacterial outer membrane? So in recent years, the Kelliher lab, alongside other research labs, discovered that a conserved lipoprotein called LPRG transports specific lipids to the mycobacterial outer membrane. LPRG is co-expressed with an integral membrane protein called RV1410C. And not only are both proteins conserved across all mycobacteria, but when both proteins are removed, decreased export and decreased surface localization of specific outer membrane lipids occurs, which suggests that both LPRG and RV1410C are important for the transport of lipids to the mycobacterial outer membrane. Additionally, although RV1410C is an integral membrane protein that is localized to the inner membrane, LPRG is actually anchored to the periplasmic face of the inner membrane as well. So due to this and the cellular location, it is therefore unclear to us how this pathway contributes to lipid transport beyond the inner membrane. And so to address this question, protein-protein interaction studies were conducted previously in the Kelliher lab in the native cellular environment by a former graduate student. And her work revealed that LPRG interacts with many proteins that are involved in many cell wall processes, including the protein antigen 85A, which is an enzyme that makes lipids that are destined for the mycobacterial outer membrane, such as this lipid, trehalose dimycolate. So due to this detected interaction between LPRG and antigen 85A, I began to wonder if the LPRG RV1410C pathway contributes to outer membrane biogenesis by impacting how well antigen 85A makes lipids. But to address this question, I first need to understand how the interaction between LPRG, RV1410C, and antigen 85A affects mycobacterial cellular functions. So to address those two research questions, Mycobacterium smegmatis was used as a model organism, and the genes for LPRG, RV1410C, and antigen 85A were removed in various combinations. Highlighted here in red are the various mutant strains that were made. And because our lab is very much interested in understanding lipid transport in the human pathogen Mycobacterium tuberculosis, the genes for LPRG and RV1410C from MTB were added to the wild-type background to produce an overexpression strain. And the MTB genes for LPRG and RV1410C were also added to the LPRG RV1410C double knockout background to produce a complement strain. And once the various complement and mutant strains were made, the lipids of these various strains were labeled metabolically using C14 acetate, solvent extracted, and thin layer chromatography was then used to assess for any changes in the presence and the amounts of the C14 metabolically labeled lipids in the various LPRG, RV1410C, and antigen85A mutant strains. So I just told you a few slides back that antigen 85A is involved in the synthesis of trehalose dimycolate. As a result of this, I first wanted to investigate if the interaction between LPRG, RV1410C, and antigen 85A is important for regulating how well antigen 85A synthesizes trehalose dimycolate. So to do this, I metabolically labeled the various strains with C14 acetate and then extracted lipids from all compartments of mycobacteria. So lipids from the outer membrane, periplasmic layer, inner membrane, and cytosol were extracted. And then these extracts were analyzed using thin layer chromatography. So the extracts are, in this instance, are called total lipid extracts. And if you look at this thin layer chromatograph on the right-hand side of the screen, you can see that in the various LPRG, RV1410C, and antigen 85A mutant strains from the total lipid extracts, it appears that trehalose dimycolate, which will be abbreviated as TDM throughout the rest of my talk, that the levels of trehalose dimycolate seems to be elevated compared to that of the wild type strain. But upon quantifying the trehalose dimycolate lipid spot, I observed that when both LPRG and RV1410C were overexpressed, or when RV1410C only was absent, or LPRG only was absent, or when both LPRG and RV1410C were absent, that the levels of trehalose dimycolate in the total lipid extracts were comparable to that of the wild type and complement strains. Additionally, when antigen 85A only was absent, or when all three proteins were absent simultaneously, LPRG, RV1410C, and antigen 85A, the levels of trehalose dimycolate was still comparable to that of the wild type strain in the total lipid extracts. So as a result, my data suggests that LPRG, RV1410C, and antigen 85A are not involved in regulating the synthesis of trehalose dimycolate. Next, because LPRG and RV1410C were previously demonstrated to be involved in the transport of lipids to the mycobacterial outer membrane, I wanted to investigate if the interaction between LPRG, RV1410C, and antigen 85A was important for regulating the transport of trehalose dimycolate to the mycobacterial outer membrane. So again, to do this, I labeled all the various mutant and complement strain lipids with C14 acetate, extracted lipids specifically from the outer membrane, and then analyzed these outer membrane lipid extracts using thin layer chromatography. And again, once I quantified the lipid spot that corresponds to trehalose dimycolate, I observed that when LPRG and RV1410C are being overexpressed, or when RV1410C only is absent, or when LPRG only is absent, or when both LPRG and RV1410C are absent, that there's actually higher amounts of trehalose dimycolate in the outer membrane extracts. Additionally, when antigen 85A only is absent, or when all three proteins, LPRG, RV1410C, and antigen 85A are absent simultaneously, there are increased amounts of trehalose dimycolate in the outer membrane extract. And so from this data, my results suggest that LPRG, RV1410C, and antigen 85A are involved in regulating how much trehalose dimycolate gets transported to the mycobacterial outer membrane. So when all of my data is put together, my research findings suggest that the interaction between LPRG, RV1410C, and antigen 85A is not important for regulating the synthesis of trehalose dimycolate, but instead is important for regulating how much trehalose dimycolate gets transported to the outer membrane. These results were a surprising find, however, as one would expect that removal of a transport pathway such as LPRG and RV1410C would lead to decreased export of trehalose dimycolate to the mycobacterial outer membrane. But instead, I observed that there's an increased trehalose dimycolate transport phenotype in the absence of LPRG, RV1410C, and antigen 85A. And so these results prompted me to ask, why does the absence of LPRG, RV1410C, and antigen 85A lead to an increased trehalose dimycolate transport phenotype to the mycobacterial outer membrane? And so one possible hypothesis is that this phenotype could be due to a general stress response. Loss of LPRG and RV1410C could possibly lead to loss of other outer membrane specific lipids, and the bacteria may be compensating for this by shuttling more trehalose dimycolate to the mycobacterial outer membrane. Or alternatively, maybe this phenotype is actually specific to the LPRG RV1410C antigen 85A interaction, right? It's possible that this pathway is responsible for transporting wild-type-like levels of trihalosyl dimycalate to the mycobacterial outer membrane, and removal of these proteins leads to a shift in some other underlying mechanism that now results in more trihalosyl dimycalate being shuttled to the mycobacterial outer membrane. Both of these questions are actively being pursued, and I hope to have experimental results at this time. But nevertheless, understanding how this pathway works to facilitate outer membrane biogenesis is important, because not only would it help us understand the steps that are involved in outer membrane biogenesis, but it more broadly could help us understand how outer membrane biogenesis might be connected to mycobacteria's ability to adapt to its environment, especially in the presence of stressors such as exposure to antibiotics and host immune responses. So I'm going to switch gears with you guys just a little bit, just to give you a very broad sense of what I hope to do for my future research work when I have my own research lab. So I'm interested in understanding how the other Mycobacterium abscessus works. Mycobacterium abscessus is a clinically relevant but critically understudied bacterial pathogen, and biofilm formation has been shown to be associated with the difficulty of clearing Mycobacterium abscessus infections. So during my PhD work, I worked on nitric oxide signaling and how it regulates biofilm formation. And currently in my postdoc, I'm looking at outer membrane biogenesis in mycobacteria. So I hope to use the technical and scientific skill sets that I've acquired from my PhD and my postdoctoral work to understand what are the molecular mechanisms that drive the biology and pathogenesis of Mycobacterium abscessus. And more specifically, I'm interested in understanding what are the molecular mechanisms that drive biofilm formation in Mycobacterium abscessus. And so with that, I would like to thank the Leading Edge Symposium Organizing Committee for the opportunity to present. I would like to thank Dr. Jessica Seeliger for allowing me to do cool and impactful science in her lab, to all the past Seeliger lab members, especially Megan Touchette, for allowing me to follow up with these LPRG protein interaction studies, to my current lab members for all of their support, especially Nitika Jaisingani for all of her expert guidance and support with all of my C14 metabolic labeling work, and all of my thin layer chromatography work, to my financial support. I'd like to say thank you. And to all of you, thank you so much for your attention, and I'd be happy to take any questions at this time.
Thank you so much, Lisa Marie. It was a very compelling talk. Very excited for your future work.
Thank you.
If there are any questions, feel free to raise your hand to ask directly, or drop them in the chat and I can ask for you. And while we're waiting for those to queue up, I'm really curious. So you mentioned that the loss of AG85A and this whole pathway both lead to upregulation of TDM in the outer membrane. And you suggested that that might be a stress response. Are there other known mycobacterial stress responses that lead to dramatic changes in outer membrane lipid identity?
Not quite. Cell envelope stress response hasn't been very well explored in mycobacteria. There are other research labs that are actively pursuing this question. So I know Tom Bernhardt's lab at Harvard, they're looking at cell envelope stress response, and they're also looking at mycolic acids, which is the type of lipid that trihalosyl dimycalate contains. So you do have that lab looking at it, but beyond that, it's not very well understood how cell envelope stress response is directly connected to lipid transport and lipid biogenesis and outer membrane biogenesis in general. But yes.
That's really fascinating. Sounds like you've identified a really interesting pathway that could be a great target for therapeutics, for sure.
Thank you. Yes.
Are there any questions from the audience? Oh, yes. Go ahead, John.
Hi, that was really cool. I got one question about what you're working on now, and then a question about your future. So I think antigen 85 is one of the antigens that people have been really working on for a long time as a vaccine candidate. I'm not a mycobacterium person, but is there any evidence that immune selective pressures from this vaccination might lead to alterations in outer membrane lipid composition and ultimately affect pathogenesis that way?
That is a great question, and I unfortunately do not know the answer to that question. I'm not sure. It's possible, but I'm not sure.
Cool. It was just a crazy question.
Yeah, no, that's a great question to think about more broadly, for sure.
For the future abscessus work, I've always thought about mycobacteria and biofilms, and so I'm just wondering, sort of, what's your system going to be moving forward? Are these biofilms thought to be sitting on lung epithelium, and will you be setting up is the thought that nitric oxide is driving biofilm formation and that it's on the extracellular surface, or are these, biofilms in vacuoles, or?
Those are great questions. So it's lung epithelium tissue culture model could be used. I know that the Eric Rubin lab, there's a postdoc who's in that lab who's actually using that system to understand a little bit more about the biology of Mycobacterium abscessus. So that's one possibility. It's not clear that nitric oxide is actually driving regulation of bacterial biofilm formation in Mycobacterium abscessus. It actually, the pathways, like the signaling pathways that are connected to driving biofilm formation is not very well understood. So I'm hoping to break some ground there. What is known for Mycobacterium abscessus is that there's at least one lipid known as glycopeptidolipids, which are affiliated with biofilm formation and affiliated with a change in the morphology of Mycobacterium abscessus. So I'm hoping to not only understand what the specific signaling pathways are, I'm also interested in possibly exploring what other additional outer membrane lipids could be important for regulating biofilm formation in Mycobacterium abscessus. Because in Mycobacteria, it's actually been shown that lipids are actually important for biofilm formation as well. So they're a little bit of the, I don't know if I want to say weird bugs, but a little bit of the interesting sort of bacteria where lipids are important, not just, a secondary messenger signaling molecule like cyclic di-GMP, which is, pretty well established in like gram-negative bacteria, for example. So, but yes, those are great questions. I hope my answers answered your question.
It was great. Thanks.
Thank you so much.
All right. Thank you so much, Lisa Marie. Very excited for your future work. And I'll hand it off to Claudia to introduce our next speaker.
Great. Thank you so much. The next speaker is Aga Kendrick. She did her PhD at the University of Colorado, Denver with Ilana Eisenmesser. And she will be talking to us a bit about her work during her postdoc in Sam Ruck-Peterson's lab at UCSD. She's also on the job market this fall. So take it away, Aga.
Great, thank you. Let me just set myself up here. Oops, I hit the wrong panel. Sorry, almost there.
No problem.
Can everyone see my slides?
Yeah, it looks great. Thank you.
Okay, great. So today I'll be talking to you about the regulation of microtubule-based intracellular transport and the work I have done to understand these processes. So to function properly, all eukaryotic cells require the microtubule cytoskeleton on which numerous cargos or cellular components are transported. And these cargos include vesicles, organelles, RNA granules, viruses, and so on, and are transported towards the microtubule plus end, which typically is near the cell peripherally by numerous kinesins, and towards the microtubule minus end, which usually is towards the cell center by cytoplasmic dynein 1. And if we look at the movement of cellular cargos within cells, as you can notice in here, these cargos move very rapidly, as shown in the human osteosarcoma cells or primary rat neurons or filamentous fungi aspergillones on the right. And I've also highlighted some specific movements in here with the arrows that show not only movement towards plus and minus end, but also rapid switches in direction, suggesting that cargos move bidirectionally. And so the fact that these cargos move bidirectionally suggests that there's opposite polarity motors present on the cargos, which is quite unique since all motors are unidirectional. So how do these motors allow for bidirectionality? And so an overreaching goal of my research is to understand how do cellular cargos achieve bidirectional movement. And specifically, I'm interested in answering the questions on how are motors regulated to allow for movement, how are opposite polarity motors move are linked to each other and to diverse cargos. And lastly, how does disruption of cargo movement affect disease progression? So today I'm going to be talking specifically about the first two questions that address the motor regulation and attachment to cargo and motors. But before I tell you about the work I have done to answer these questions, I want to just give you a little more background on what do we know about motors. So all the plus and directed transport is carried out by the kinesin superfamily, and kinesins can be auto-inhibited. And then upon release from auto-inhibition, kinesins can either bind to cargo directly or via adapter proteins. All the minus and directed transport is carried out by cytoplasmic dynein 1, which is a large complex of 12 proteins. Dynein is also auto-inhibited, but once relieved from auto-inhibition, dynein assemble into this even larger complex with its co-activator dynactin and an activating adapter protein. And this activating adapter is what links dynein to cargo either directly or indirectly. So to answer how motors are regulated, I first focused on the dynein mechanism of regulation. And as I mentioned, dynein is auto-inhibited and appears in this biparticle conformation. And so recent work from our lab and others suggests that this protein LIS1 could be involved in relieving the auto-inhibitor mechanism of dynein. And LIS1 is particularly interesting as mutations in LIS1 and depletion of LIS1 lead to this very severe neurodevelopmental disorder, lysencephaly. LIS1 is also the only dynein regulator that binds directly to its motor domain, as shown by these structures from our lab. So what do I mean by dynein motor domain? So in order for dynein to move on microtubules, there has to be ATP hydrolysis that takes place in dynein's motor domain, as highlighted here in color. So if we just focus for a little bit on the motor domain itself, I marked the different ATPases in here. So dynein is a member of the AAA family of proteins that's composed of 6 hexameric ATPases, and ATP AAA 1 through 4 are the ones that bind to ATP. And so upon ATP hydrolysis, dynein is capable of forming rearrangements within a ring, linker movement, and then movement on microtubules, as shown by this oversimplified schematic in here. And then we know from work from our lab that ATP hydrolysis in dynein's ring influences the way LIS1 binds to dynein. But our previous work has been done with either mutated dynein constructs or with non-hydrolyzable ATP analogs. So I really wanted to know what happens during the different steps of ATP hydrolysis, and can we identify the intermediate states of ATP hydrolysis and LIS1 binding to really understand what is the function of LIS1 in dynein. And so to do this, I used cryo-EM to capture different states of dynein mechanochemical cycle. And I purified yeast dynein, and then I pre-incubated it with or without LIS1, and then made cryo-EM grids within 30 seconds of ATP addition with the hope that I can capture the different states of binding. And that's exactly what I did. So from this analysis, I was able to identify 9 different structures, and I colored these structures based on the schematic on the left. So you can see the different AAA domains as well as the linker. And hopefully you can appreciate in here that all these structures have different conformations and show different LIS1 binding. I also had a very good resolution within the AAA rings. So I was able to assign the nucleotide states for the 4 AAA domains that bind nucleotide. And one interesting aspect of this data was that if I compare the number of structures I generated from the data set that I collected without LIS1 present, I only got 1 structure. While in the presence of LIS1, I was able to identify 8 different structures that all come from the same data set, suggesting that LIS1 may be stabilizing the different steps of dynein's mechanochemical cycle. So if we look at these structures in a little more, or these cryo-EM balls in a little more detail, I grouped them all together in here to show you the conformational changes that take place within the dynein ring and with LIS1 binding. So this really captures some steps of dynein's mechanochemical cycle. So based on my data, I propose a model in which the phi particle could be open in the following way. So one of my structures is consistent with the previously solved structure of the phi particle within just a motor domain. And so I would propose that upon ATP N+1 addition, dynein ring rearranges itself to allow for 2+1 binding. And I haven't really talked about this, but I'm also working on the project in the lab where we identify a new structure called this, which we call chi particle, that corresponds to 2+1 being bound and a slight opening of this phi particle. And then during further steps of ATP hydrolysis and LIS1 binding, this can further lead to opening of the phi particle, release of one of the LIS1, and then an eventual removal of LIS1 and assembly of the dynein complex that is able to move on microtubules. So I've shown you today how our motors regulated on via the example of dynein, and I don't have time to talk about this, but I've also investigated regulation of kinesin 3 KIP1C. It's shown that this is an active motor. But what I want to focus on for the last couple minutes of my talk is how our motors link to each other and to diverse cargo. So to answer this question, I use proximity proteomics approach when I looked at the hook family of activating adapters and identify different cargo adapter complexes, a family of different cargo adapter complexes. And my analysis also revealed different cargos that these complexes are linking to. And using live cell imaging and biochemistry, I was able to confirm these interactions as shown for RAB5 marked early endosome movement on microtubules on the left and RAB1 ER2 Golgi tubules on the right. And this analysis also identified kinesin and KIP1C, its possible interaction with activating adapter hook 3. So I used biochemical and cell biology approaches to also confirm that interaction. But this would suggest that maybe hook 3 could scaffold dynein and kinesin for bidirectional motility. So to test this, I use in vitro reconstitution with purified components and TURF microscopy, and shown that in an in vitro system, dynein and KIP1C are scaffolded by hook 3 for bidirectional motility, as shown in these movies where microtubules are in blue and the motors are either in magenta or green. So what I've shown you today is some mechanisms of motor regulation, as well as how motors could be linked to each other and to cargos. But several questions still remained, and I plan to explore these questions in my future lab. So my proteomic analysis identified other cargos that I didn't really have time to explore, for example, mitochondria and endosomes. So in my future lab, I plan to identify the connections between these cargos and the cargo adapter complexes that allow for dynein to attach to these cargo. This analysis also identifies several different kinesins that are possibly linked to these cargos, providing a link to bidirectionality between these cargos. And bidirectional movement can also be achieved by movement on other cellular tracks, for example, actin. And indeed, if I look back into my analysis, I can see actin motors, myosins also present in my mass spec data set. So exploring these interactions will be the next steps in my future lab. And then lastly, several of the components of these complexes carry disease-causing mutations. So in my future lab, I plan to explore how does disruption of cargo movement affect disease progression. So with this, I would like to thank the Reg Peterson lab, especially Sam, for all her help, my collaborators present and past in the lab. I've also worked with some really great undergrads who have been fundamental to keep these projects moving. All my structural work that I presented has been done in collaboration with Andreas Leschinger lab. And then I would like to thank my funding and also Leading Edge fellows and Cara for giving me this opportunity to talk and creating this amazing supportive environment. And with this, I'll take questions. Thank you.
That was an awesome talk, Aga. Thank you so much for sharing your work with us. So if anyone has any questions, please raise your hand or put them in the chat and I'll ask them. I I had a question about kind of these dueling motors and how they might be, like how you think it might be set so that they go one direction versus the other. Like one idea is that you have more of like a kinesin and that drives it with the plus end, and then you have more dyneins, or is it that like it has more to do with kind of these adapters that activate or inactivate? Or I'm just kind of curious your thoughts on that.
Yeah, so that's a great question, and there's a lot of research in the field trying to explore these questions. There's definitely some studies showing that teams of motors can, so there's definitely teams of motors on cargos based on what we know so far. And there's some studies, usually biophysical studies, showing that teams of motors can move cargo, so they can kind of engage in these tug of war between different motors, but we really actually don't fully understand how that happens. And the idea of cargo adapters to regulating this system is not very well explored too. And there's some cases also where only one motor can engage at a time. So, I mean, that's a really good question. I don't have a full answer for it. I don't know that we really know, but that's definitely something that I want to explore in the future.
Great. Thank you so much. Yeah, I think that hopefully that question is in really good hands now. So I'm excited to see what happens with it. So in the interest of staying on schedule, I'll pass it off to Zara to introduce our next speaker.
Great, thanks for a lovely talk, Aga. It's my pleasure to introduce Priyanka Joshi, who did her PhD work at University of Cambridge in the Ventricular Lab, where she then went on to receive a Marie Curie Fellowship to do independent postdoctoral work at Cambridge, and is now a research fellow at University of California, Berkeley. Priyanka is going to talk to us today about some awesome cell biology. Priyanka, why don't you get started?
Thank you, Zara, for that introduction. Can you see my screen?
Yep.
Okay, great. All right.
Yeah,
thanks. Hi, everyone. Today, I'll be talking about my research on the mechanisms of biochemical homeostasis in aging and age-related diseases, essentially a balance between micromolecules, metabolites, and proteins, the macromolecules. So a lot of my work has been inspired by this book called Biochemical Adaptation by George and Sumero that I read, that states that life is fragile because it also must be efficient, accurate, and responsive. And a lot of these studies have been inspired by fishes in different environmental, different geographical habitats, where essentially they adapt to the depth and the temperature, not exactly by intrinsic adaptation, which is by chemical changes in the protein structure, but actually by extrinsic adaptation, meaning that by the changing levels of micromolecules. So micromolecules are essentially in the media. So if you look at the cell as an aqueous solvent with water, any kind of alteration in this media that bathes the large molecules that can adaptively modify it and change the stabilities and functional properties of the proteins. Now, these extrinsic adaptations can actually also be because of other environmental factors, such as exercise, temperature changes, diet, various kind of pathological sources, or aging, or just stochastic processes. And this is what led me to think about if there can be other factors, environmental factors, that can change the structure of proteins. Now, if you look at proteins, because of the intra and intermolecular interactions, they can move from their native form to liquid. They can phase separate and form the droplet form, or mature into very stable amyloid-like fibrils. Now, in certain diseases, such as Alzheimer's, Parkinson's, and ALS, you see that these protein aggregates start from one part of the brain and reach out to the next, to the other forms, and all of them have this underlying commonality, which is these protein aggregates. But how they start from one part and progress to the other is not known. And one of the things that is very compelling is that a lot of these diseases, they start much earlier on than they present themselves in the clinic. That is, the aggregation process starts in the brain very, very early on. And that is like life is always on the edge. As we had seen that in the cell, you have proteins, you have metabolites, which are like small molecules, and a whole lot of other small inorganic ions as well. There have been studies that show that the proteins, they are metastable and they are at an edge. So there is a correlation between their expression levels and their aggregation, meaning that even if there is a slight disruption because of any kind of external stimuli, they might aggregate. And I have hypothesized that such a thing can also exist in case of metabolites, where they also exist in within a threshold, and because of environmental stimuli, they can also compartmentalize and form either membraneless compartments or aggregate. So with this, my research vision has been largely how to understand that how changes in the physicochemical microenvironment such as pH, ionic strength, can lead to molecular crowding. That is how proteins, I asked the question, how proteins respond to this change as a function of age, as an environmental stressor. I combine the fields of biophysics and biophysics of protein aggregation and metabolism in the context of healthy aging and age-related diseases. And I use the model C. elegans because of its short lifespan. It lives for about 2 weeks. Its genetics is largely known. And it's a very powerful model to study aging. And I also use neuroblastoma cells towards, all in all, towards the biophysical determination of cellular and organismal aging. Now, how it all started was like, for my graduate work, I was designing small molecule inhibitors of protein aggregation, where I started with small compounds that were already known to bind monomer, oligomers, and fibrils. What I did was I took these inhibitors, broke them down into small fragments, F1, F2, F3, F4, and then recombined them and searched various databases to find for novel compounds that could inhibit protein aggregation. Now, this made me think that if we can drug these protein aggregates, then nature must have come up with a source. Evolution must have come up with it on its own. And it could be in the form of small endogenous metabolites, which already exist in the cells. So with this, I'll be presenting 3 of the studies today, which tie together, which is my postdoctoral work, which tie together this work on metabolites and protein homeostasis. I'll talk about this story on the first story about this, how metabolites induce the cytosolic unfolded protein response, and then how they actually act directly on protein aggregates. So in this story, we looked at the hallmarks of Alzheimer's disease, as they are A-beta plaques and tangles, but there are a whole lot of biochemical processes that are dysfunctional. And what is very interesting is that all of these biochemical processes have small molecule metabolites, these endogenous metabolites. So the hypothesis here was that if they are imbalanced in Alzheimer's disease, then we could rebalance this protein homeostasis by using these small molecule metabolites as proteostasis regulators, which alongside chaperones and other detoxifying enzymes and clearance mechanisms can balance it out. So what I did was I took some small molecule proteostasis regulators from Rick Morimoto's lab and applied the same strategy that I had developed during my PhD work, which was to calculate these fragments in drug-like molecules, found the similarity scores, and ran that on the human metabolome database, and identified metabolites that were either upregulated or downregulated, and took this pool further for experimental validation. 6 of these metabolites were actually proteostasis modulators, and we took these to test them in worms. So in C. elegans, at day 5, there is a GMC model where you express A-beta 42 in the muscle cell, and as it aggregates, it causes worm paralysis. And what I saw was that 2 metabolites, carnosine and kynurenic acid, were able to rescue this phenotype by improving fitness in the speed, the bends per minute, and the live ratio, as you can see on the left-hand side panel, but did not have any effect in the control. What I saw was in the worm head, they also reduce the aggregates. So here, the orange aggregates that you can see are stained by NIAID4, which is a dye that preferentially binds to the beta sheet of the amyloid. And here you can see that after you give carnosine and kynurenic acid, which is on the lower left-hand two panels, you can see that it basically rescues and removes these aggregates. And so we were able to see that carnosine and kynurenic acid reduce the aggregates, A beta aggregates. So essentially, suggesting that metabolic dysregulation can lead to changes in metabolites. And here, these metabolites can induce the heat shock factor 1 and downstream chaperones, and which can increase protein folding and decrease protein aggregation. And overall, this was I was able to show that this is through the DNJ12 and DNJ9. 19, which are the J factors chaperones, which are also downstream of HSF1. So then I looked back at my series of metabolites, and I wanted to see what could be the other metabolites that we can test that modulate aggregation. In this screen, I looked at vitamins. And the whole idea was to see the microscopic steps at which these vitamins may affect aggregation. So A beta aggregation can proceed by primary nucleation, which is the monomers bind to each other and they form oligomers and eventually fibrils, or they can have secondary nucleation or elongation. And metabolites can actually give the structure, give a surface by which this aggregation takes place. So in a vitamin screen, I identified vitamins that delay aggregation or accelerate aggregation. And in these two, I identified vitamin A that inhibits aggregation and vitamin E that accelerates aggregation. Using these microscopic steps and fitting these equations, I was able to identify that vitamin A inhibits the aggregation by affecting both primary and secondary nucleation. So far, people have studied vitamins in this context, but nobody has looked at how microscopic effects are affected. And that is very, very crucial for understanding the mechanisms of how macromolecules are affecting protein aggregation. In case of vitamin E, I was able to see that vitamin E by itself is accelerating aggregation on its own. But if you break it down into monomeric forms, it is able to delay aggregation. And what was most interesting was that if you combine the two in a certain range, you were able to completely nullify this effect. So in this phase diagram, you can see that a certain range of concentrations, which is the white zone, the effects of both the metabolites, both the vitamins was nullified, which suggests that there could be a protective role, which by the simultaneous presence of aggregation inhibitors and accelerators. So this is essentially a proof of principle, a systematic approach to investigate how we can take dysregulated metabolites and study them on aggregation processes. Now I've identified these 4 metabolites: carnosine, kynurenic acid, vitamin A and E. But as one may think about or ask this question, that vitamins, that metabolites can have offset effects in the cells as well. So for this, I use limited proteolysis-based mass spectrometry, which essentially identifies any conformational changes in the whole cellular proteome. So here I used neuroblastoma cells and applied a range of these metabolite concentrations, and then did limited proteolysis, and we could find out that there are certain conformotypic peptides that result in the as these metabolites are binding. So essentially, we could identify the targets. And what was most interesting was that these dysregulated metabolites induce conformational changes in the proteins that were associated with protein misfolding diseases. And the molecular function, the most top ones were a protein folding chaperone, suggesting that when there is any environmental change, probably the levels of metabolites are dysregulated and fluctuate, and they can have an effect on the conformational change. So together, these 3 studies were able to bring together these pieces of this jigsaw puzzle, where I show that metabolites can affect protein homeostasis and protein aggregation. Right now, I'm also working on the mechanisms of calorie restriction, where calorie restriction is the most robust mechanisms for lifespan extension. And that also involves certain metabolites like ADP, NAD, but we do not really know how it affects protein aggregation. So something that I don't talk about now in this, the scope of time, but I'm studying this in C. elegans as well. So in my lab, I would like to understand the mechanisms of how unfolded proteins form of misfolded proteins, and this essentially study the balance between proteins and metabolites and how this balance gets disrupted due to aging and causes disease. The big question that I've asked is, what are the mechanisms by which metabolites function to maintain protein homeostasis in cells, tissues, and ultimately organisms? And how I plan to solve this is, one is to investigate the role of vitamins, polyphosphates, and sugars as chemical chaperones. There have been certain osmolytes that have been studied as chaperones, but I would like to investigate these three sets of metabolites by using thermal unfolding and also cryo-EM that I have done in the past at Cambridge in the Department of Biochemistry. In the second approach would be the in vivo manipulation of metabolites to see if they can affect aggregation or biomolecular condensate formation. And for this, I would be using a limited proteolysis-based mass spec, and also some of the machine learning-based lifespan machines that I have set up at UC Berkeley in the TTOV lab at this point. The third approach would be to develop high throughput methods to study the mechanisms of amyloid formation. So far, we have studied how proteins form amyloids, but these small molecules, if they are aged in the test tube or they can also form amyloids, but this is not studied very well. So using fluorescence-based methods, such as the ones I have studied before for protein amyloids and cryo-EM, I'd like to study a metabolite amyloid formation. With this, I'd like to thank first of all the Cambridge, Chris's lab, the Dobson lab, and Michele's lab, where I studied, where I learned all the tools of the biophysics of protein aggregation. And from there on, I collaborated with Paola Picotti's lab at ET at Zurich, where I learned how to do limited proteolysis-based mass spectrometry. And combining this, I was very, very interested in how metabolism and aging affects protein aggregation. So I chose to move to Berkeley in Dennis Titov's lab, where I'm looking at how calorie restriction affects lifespan and health span. And throughout, my mentors have been Rick and John, who have advised me on developing C. elegans to answer the question that I'm interested in. I'd also like to thank Downing College for my independent space to conduct this work, Marie Curie Fellowship, and Biochemical Society, and also Leading Edge for providing this platform to share with all of you about my research. And I'm open to questions.
Thank you so much, Priyanka, for that incredible work that you just described. In the interest of time, I think we're going to move on to our next speaker, but if you have any questions for Priyanka, please feel free to drop them in the chat and she'll be able to respond there. All right, Claudia, would you like to introduce our next speaker?
Yeah, great. Thank you so much, Priyanka, for that really great talk. And so our last speaker of this session is Jess Kelliher. She did her PhD at the University of Illinois Urbana-Champaign in the Cal-Phi lab, and she'll be talking about her work in the Sauer lab at the University of Wisconsin-Madison. Take it away, Jess.
Awesome. All right, can you see presenter view or my slides?
We can see, it looks great.
Okay, great, awesome. Okay, let me start by saying thank you so much to Leading Edge, especially to our wonderful moderators today, and to Kara for the opportunity to present my work. So I am really fascinated by host pathogen interactions that occur in the cytosol. And so today I'm going to tell you a brief story about how a professional cytosolic pathogen is able to resist cell wall stress in the cytosol. And then I'll tell you a bit about my future interests for the Kelliher lab in the future. So the way that I think about my work is through the lens that pathogens are adapted to respond to stress in their particular infectious niche. So for example, extracellular pathogens like Staphylococcus aureus respond to stressors like the complement system or antibodies. Vacuolar pathogens like Legionella have to contend with lysosomal fusion and an increasingly acidic pH. So in the case of bacterial pathogens that infect the cytosol, we actually have a less clear understanding of the host defenses that are present in this niche. But we do know that the cytosol is restrictive because bacteria that are not adapted to this environment are killed. And so one of my major research interests is in understanding how the cytosol can restrict the growth of non-adapted invaders. And because I'm really a bacterial physiologist at heart, I'm also equally interested in understanding how professional cytosolic pathogens are able to adapt to this niche. So to model the cytosolic host pathogen interface, I used the organism Listeria monocytogenes, which is a formidable foodborne pathogen, but it's also an excellent model organism for studying host pathogen interactions in the cytosol, cell biology, and more. And so the life cycle of Listeria, just briefly, once Listeria is taken up into a host vacuole, it escapes the vacuole and reaches the cytosol, where it replicates and then spreads cell to cell. And so Listeria really requires the cytosolic phase of its lifestyle in order to cause disease. So one factor that the Sauer lab previously determined is essential for Listeria's ability to adapt to the cytosol is PERK-A, which is a member of the pasta kinase family. These kinases are comprised of extracellular pasta domains that sit in the peptidoglycan layer, which is the major component of the bacterial cell wall in gram-positives like Listeria. And these pasta domains, when they sense cell wall stress, they propagate that signal to a eukaryotic-like serine-threonine kinase domain in the bacterial cytoplasm. Because these are serine-threonine kinases, they phosphorylate multiple targets. And in other bacteria where pasta kinase targets have been identified, they include proteins involved in not only cell wall homeostasis, but also central metabolism, other stress responses, and more. And so in this way, we really consider pasta kinases to be master regulators of cell wall stress in bacteria. And Lisa Marie gave us a really great introduction into the importance of maintaining the cell envelope for pathogenic bacteria, for resisting cell wall stress, or for resisting stress in the host, as well as for resisting antibiotic stress. And so we know that pasta kinases, or the pasta kinase in Listeria, is important for resisting cell wall stress in vitro. So this is an MIC experiment where I've exposed wild type and a PERK-A mutant to increasing concentrations of ceftriaxone. And we see that the PERK-A mutant is about 100-fold more sensitive than wild type to this beta-lactam antibiotic. As I mentioned, the PERK-A is also required for Listeria's ability to adapt to the cytosol. So while wild type is able to replicate in the cytosol of macrophages, a PERK-A mutant is restricted and even cleared in that same time frame. And a PERK-A mutant is avirulent in a mouse model of systemic listeriosis, really highlighting the essentiality of this regulator for the pathogenesis of Listeria. So when I began working on this project, I became really interested in understanding the mechanisms through which PERK-A is promoting adaptation to the cytosol. And as a first step to understanding this, I comprehensively identified PERK-A targets in the context of cell wall stress using phosphoproteomics. So I grew wild type and the PERK-A mutant in a sub-inhibitory concentration of the beta-lactam ceftriaxone and compared the phosphoproteomes of both these strains to identify proteins that are specifically phosphorylated by PERK-A. And we found that PERK-A phosphorylates 23 proteins, and I don't want you to read this list or memorize it, but I do just want to point out that these PERK-A substrates are involved in diverse processes, from maintaining the cell wall to oxidative stress response, metabolism, and more, really consistent with the idea that PERK-A is a global regulator of physiology in response to cell wall stress. So today I will briefly follow up on one of these targets named REOM, which is a conserved protein among gram-positives that contain the pasta kinase. So REOM is a small protein that interacts with MUR-A to target it for degradation through the CLIP-CP protease. MUR-A is an essential enzyme involved in cell wall synthesis, and so the presence of REOM has a negative impact on cell wall synthesis overall. The same group that first discovered REOM in Listeria also showed that it can be directly phosphorylated by PERK-A, and PERK-A phosphorylation of REOM prevents its interaction with murA, and this results in an increase in murA protein levels. So I wanted to test the hypothesis that during cell wall stress, this phosphorylation of REOM is important for increasing peptidoglycan biosynthesis. And so to test that, I used a click chemistry-based peptidoglycan labeling approach where I grow Listeria to mid-log phase, and then I add a clickable PG precursor and induce stress or not, and grow bugs for another hour. Then I perform a click reaction to conjugate a fluorescent molecule to that functional group on the PG probe. And then I can perform flow cytometry to quantify total fluorescence as a measure of how much nascent peptidoglycan synthesis has happened in that hour. So using this technique, I found that when I induce cell wall stress, wild type increases peptidoglycan synthesis about twofold. In contrast, a PERK-A mutant, which has similar basal levels of PG synthesis, is unable to increase PG synthesis during stress, suggesting that PERK-A is important for that boost in PG synthesis we see in wild type. On the other hand, an REOM mutant has constitutively higher peptidoglycan synthesis, even in the absence of stress. And this is consistent with the idea that we've removed that negative impact on murA protein levels. And if we delete REOM in a PERK-A background, we can completely restore peptidoglycan synthesis in that strain, suggesting that PERK-A is phosphorylating REOM in order to increase peptidoglycan synthesis during stress. So to test whether this pathway is important for Listeria's ability to respond and grow during cell wall stress, I performed another ceftriaxone MIC, where again, a PERK-A mutant is much more sensitive. And we see that if we delete REOM in a PERK-A background, we can almost completely restore intrinsic resistance to ceftriaxone, indicating that this pathway is important for responding to cell wall stress. So I was next really interested in the question of whether this PERK-A regulation of REOM is important for adaptation to the cytosol. And to ask that question, I used an assay that we've developed in the lab that specifically measures killing of Listeria in the cytosol of macrophages. So in this assay, strains of interest are carrying a plasmid that encodes a luciferase gene under the control of a host-driven promoter. Strains like wild type that are adapted to the cytosol retain the plasmid, but strains that are sensitive to cytosolic stressors are killed. And upon lysis, they release the plasmid, which can be taken up and expressed by the host. And then we measure light as a proxy for bacterial killing. So in this assay, a PERK-A mutant is killed at rates 10 to 15 times higher than wild type. But remarkably, deleting REOM in the PERK-A background completely restores resistance to the cytosol of the PERK-A mutant, indicating that this pathway is contributing to the ability of Listeria to adapt to stress in the cytosol. So next, to test the hypothesis that this pathway is important for the pathogenesis of Listeria, I turned to our mouse model of systemic listeriosis, where again, a PERK-A mutant is avirulent. Interestingly, we saw that an REOM single mutant has a virulence defect. And this suggests to us that constitutive overactivation of PG synthesis is also detrimental to Listeria's ability to cause disease. But deleting REOM in a PERK-A background almost completely suppresses the virulence defect of this mutant, indicating that this pathway does contribute to pathogenesis in Listeria. So just to summarize this story briefly, I've shown you that during cell wall stress, both in vitro and in the cytosol, PERK-A is phosphorylating REOM in order to increase peptidoglycan synthesis. And so this pathway we've shown is clearly important to the pathogenesis of Listeria, but PERK-A phosphorylates many other proteins. And so moving forward, I'm interested in understanding how these other substrates are contributing to adaptation. And so one, if we come back to this list of phosphoproteomic targets, one class of substrates that I'm really interested in is these other two component systems that both of which sense cell wall stress in Listeria. So I'm interested in understanding how a combination of different cell wall sensing systems in Listeria are able to contribute to resisting cell wall stress in vitro and in the cytosol. Another really interesting observation that I didn't have time to touch on at all today is that a PERK mutant also seems to have a defect in the ability to produce and secrete virulence factors that are required for escaping from the vacuole and spreading cell to cell. And so I'm really interested in understanding how PERK is involved in activating, directly the virulence program of Listeria, which I think is, I hypothesize, is mediated through this phosphosubstrate cis-K. And then as a balancing act, there's a phenotype with REOM mutants where we see a defect in secreting virulence factors as well. So we think that PERK is modulating cell wall synthesis and virulence factor production. And so I'm interested in understanding those competing processes. And then lastly, I'm really interested to know what are these host vectors in the cytosol that are causing Listeria to experience cell wall stress. And towards that end, I'm performing a parallel CRISPR screen in macrophages to identify pathways that are causing cell wall stress in Listeria, as well as a proximity labeling approach to identify host proteins that are directly interacting with the Listeria cell wall in the cytosol. And so with that, I'll just quickly acknowledge the Sauer Lab, especially JD Sauer, who's a fantastic mentor for postdocs, as well as the Sauer Lab members listed here who have contributed directly to this project. Thanks to collaborators and funding. Thanks again to the Leading Edge Symposium for the opportunity to present my work, and I would be very happy to take any questions.
Awesome. Thank you so much, Jess. That was a really beautiful talk. And again, if anyone has any questions, please, if you want me to ask them, please put them in the chat, or if you want to ask them directly, raise your hand and I'll ask them. And as we wait for any questions there, I was curious about what you might think, like you're doing this, setting up potentially the screen in macrophages to look at cytosolic factors that affect bacterial cell wall stress. And I was curious if there was like how, like does Listeria have different pathogenicity in different cells? Like could different cytosols be like more or less stressful, and that's why Listeria try to get into some cell types versus others? And if that's kind of, if you have any ideas on that.
Yeah, that's a great question. And it's something that we think about a lot, actually. And I'm definitely interested in exploring as I'm thinking about setting up these screens and this labeling approach. So we do think that different cytosols have different restricting factors for Listeria. And the reason we think that is that using our cytosolic killing assay, where we measure plasmid release and luciferase production, we see that mutants of Listeria that are sensitive to different in vitro stressors, such as oxidative stress, are sensitive to some cytosols. For example, they're sensitive to killing in the macrophage, but not in epithelial cells. So we definitely think that there are likely different antimicrobial pathways and factors in these cells. I will say that PERK is kind of great because it's sensitive to every cell type that we've tested it in, which is consistent with its role in sort of affecting physiology in a variety of ways. But yeah, that's definitely something we're interested in investigating.
Great. Thank you so much. Brooke Gardner has a last question. Is there a phosphatase that opposes PERK activity? That's clever.
Yes, there is a phosphatase. Something I didn't touch on is that postakinases are conserved in gram-positive, so in firmicutes and actinobacteria, almost exclusively in single copy, which is really interesting. And then I think pretty much universally, there's also a phosphatase encoded at the same locus. And depending on the organism, some people have studied the role of the phosphatase as well as the kinase in resisting cell wall stress and in these different pathways. That's not something I have explored yet, but it's definitely on the list of things to do, is to understand how the phosphatase also contributes to these environments. Thanks.
Great. Thank you so much, Jess, and to the rest of the speakers in this great cell bio section, Leading Edge Symposium. Thank you all to everyone who listened and tuned in. We'll have the last two sessions of the symposium on Wednesday and Friday. So tune in then and have a good rest of your days.